I am Tony Patalunas. I'm a program manager at Seven Bridges, working on BioData Catalyst, and I am joined by my colleague, Dave Roberson, who is our community engagement manager. And as Amber noted, we're going to cover interactive analysis today. Next slide. So we can start, before I start, let's talk about the key takeaways for today, what we're hoping that you walk away with after the session. So we are hoping that you learn, if you haven't learned yet, how to access RStudio, JupyterLab notebooks, and SAS Studio. So what we would refer to as the interactive environments on Seven Bridges. We want to make sure that you understand how and when to select what kind of resources you need for your interactive session, whether you need CPUs or GPUs, how much RAM, how much storage you need. We want to go over how to upload your own script if you're working locally or on an HPC, how you can move that into the cloud and work on it in Seven Bridges, and then how from the interactive analysis, you can access your data sets, whether it's controlled data that you have access to, such as the hosted TOPMed data, or your own private data that you've uploaded to Seven Bridges. And we're going to do all of this in the context of GWAS with the Genesis pipeline. So that's primarily for Dave's demo after my handful of slides here. So we can start by defining interactive analysis. And there's really kind of two categories of analysis that we'll talk about. Interactive analysis is immediate real-time feedback on whatever analysis or whatever you're doing. And it's typically iterative in a fast way. So like the GIF that I have here, you can see I'm trying to install a package in RStudio, and I'm getting immediate feedback of errors. This is juxtaposed to submissions or jobs batch analysis, where you're waiting for feedback and iteration. So you'll submit something to a queue and wait for feedback. We're focusing on interactive analysis today. And so you might be familiar with a high performance computing cluster at your local institution or your local environment, but on an HPC, if you're working on an HPC, you might be familiar with Slurm. Slurm is a very common job handler for HPCs. And so interactive analysis on the HPC is typically done either through srun or sinteractive. And that's where you log in on the head node and you'll use srun or sinteractive to go to another node, a working node. You typically have a small amount of resources. Maybe you're looking at 10 gigs of RAM, something like that, maybe 8 cores. And that is, again, compared to batch analysis, which is normally when you want to scale up. So say you want to reserve a whole node, 50 cores, 256 gigabytes of RAM, you're going to use something like sbatch or job submission. So again, the comparison for the HPC is sinteractive versus sbatch job submission. And we can bring that same comparison to the cloud. Next slide. For interactive analysis versus batch analysis. So interactive, I had said, it's immediate feedback. It's iterative, so it's like running a notebook. You can open an RStudio notebook, JupyterLab's notebook, and run line by line code if you want. Same thing with writing a script. So if you're prototyping or developing a new script, testing a new package that you found and you're curious about. And we also put our Shiny tool analysis in this category because that is also for visualization and live feedback and immediate iteration. Again, we'll draw the comparison to batch analysis, and in the cloud, it's typically running individual tools or workflows. Workflows are tools that are strung together. And tools in the cloud here, we define as workflow language wrapped scripts or applications. So Seven Bridges primarily uses CWL, the common workflow language. But starting in February, you can email support and request access, early access to WDL and Nextflow on the Seven Bridges platform if you're interested in trying those out. And so when would you use interactive analysis versus the batch analysis? So times when you need immediate feedback is when you want to use interactive analysis. And some examples are harmonizing data. Perhaps you have access to a bunch of different TOPMed studies and you need to glue some together, harmonize them. We'll cover a live demo of this. Prototyping a new script. Like I said, you found a new library. Very cool. You want to try it out. You're going to build a new script around it. You want that immediate feedback. Visualization of data. So you can use RStudio or JupyterLab's notebooks or our Shiny tools to pop up graphs and get live visualization to really understand your data better. In debugging or troubleshooting, think about one of your lab mates or a collaborator or colleague sends you their script and you need to debug it or troubleshoot it for your own data. Generating summary statistics is another key place where interactive analysis is very important. So you have your data frame and you want to look at, just the general spread, mean, median, mode. You want to understand what's there and maybe plot that again to get a good understanding of data. You want to do that iteratively and on a live basis instead of submitting and waiting for it. And then creating markdown reports. So either R markdown language or markdown in Python, where you're annotating code, maybe you have embedded graphs. You can print those out as PDF or HTML so that you can share them with colleagues or tutorials, maybe in your own Git repo. And so on 7Bridges, BioData Catalyst powered by 7Bridges, we have 3 interactive environments here. The back panel here is the RStudio environment. It looks exactly like RStudio on your local machine. So you have the same boxes here with your script and your command line, your environmental variables, and your file picker, which you can't see. The middle box here is the JupyterLabs notebook, and you have access to Python 2, Python 3, Julia, and the command line terminal, as well as a text editor if you want to write scripts or just write notes, files, readmes, etc. And the third environment is SAS Studio. This again looks exactly like the SAS Studio you might be familiar with on your local laptop. Today for the live demo, we are going to cover the RStudio and the JupyterLabs environments, specifically Python. We're not going to cover the SAS Studio environment, but we included a button on here for the View SAS Guide. We have a SAS guide written. If you are interested in working in SAS and want to get on there, Amber had posted the slides in the chat, so you can just click on that button and it'll take you to the 7Bridges guide for SAS usage. And so we're going to go into the live demo with a case study, right? So let's pretend I'm a researcher from a university medical center and I have 200 patients whole genome and phenotype analysis. So I'm looking to run a GWAS for the chronic condition that I am studying, HPC to BDC. Okay? And I started building my GWAS script locally, and I realized this isn't the best. I'm not going to run this on my laptop, right? I got 200 patients. That probably won't run on my laptop, but I need to scale up. I need more control data. And I found in the TOPMed Freeze 8 data, there's a great control population. So what I need to do is get on BioData Catalyst, move my script onto BioData Catalyst. I need to finish building that pipeline and test it. So that's where interactive analysis comes in to building my script. I'm going to need to harmonize the data. So between my 200 patient genotype samples and the TOPMed Freeze 8 data, I need to harmonize that. I'll need to run quality control on the genotypes here. And then eventually I'll scale up my analysis with the power of the cloud. And so that's where the batch submission comes in, right? We iterate and we prototype, and then we submit to the batch analysis in the cloud. And so before I turn it over to Dave, I just want to note here that we have another button that is the guide to GWAS with the Genesis pipeline. So if you are interested in deep diving into this beyond whatever's shown today, you can read through or even perform this guide on your own. So at that, I will turn it over to Dave to go through a live demonstration.
Thank you, Tony. And I hope that we can make headway to your HPC to BDC chronic condition. It sounds fascinating. Okay, so let's go ahead and get started. Once again, thank you for the introduction, Amber and Tony. So what I'd like to show you is interactive analysis in the use case of doing a genome-wide association study. So we're just going to lightly touch on some of the things that Tony just outlined. I've already made this project on BioData Catalyst powered by 7Bridges. And in the description, I took some notes about what we want to accomplish in the next 20 minutes. So I created 3 objectives for us. One, we want to show you how to upload a script quickly through your web browser that you may be working on locally or on your HPC. That's basically a text file, so you can upload it very quickly, but there are certain things you might want to consider after you upload it. Next, we're going to demonstrate how to do interactive phenotype harmonization via RStudio. And finally, end with genotype quality control via JupyterLab. And you can see how we broke it up. We wanted to show you 2 separate environments, depending on if you're an R person or a Python person or somewhere in between. SAS is also available, but we'll not be demoing that today. Really quickly, The materials for this are also available to you. So you'll have the slides, but if you want to dive deeper, this is based on 2 public projects you can get to. So anyone that has an account, if you go to public projects and you find the Genesis tutorial, this was created by the University of Washington Genome Analysis Center, and it's actually what they use for their Summer Institute in Statistical Genetics for one of the modules. So it's really in-depth. I'm going to show you part of that. So that's in the public project. And the other part I'm going to show you is quality control of a VCF. So this would be the genotypes that you need for your single variant association test. You can find that notebook in the public project called Data Cruncher Interactive Analysis. And it's actually a series of interactive notebooks that do a whole bunch of different things. If you don't know, public projects are completely worked out analysis that you can sort of look through, or you can copy the entire project and start to edit some of the parameters. So let's get started. Everyone should know by now, like the general outline of what a project is and how you collaborate. Along the top, you can access your files, your apps, and your tasks. But we're focusing on interactive analysis today, which is all the way on the right side. So if I click on that, it brings up the different types of interactive analysis we have. So there's a genome browser, which you can view BAM or CRAM files. There's a card for Data Cruncher. So Data Cruncher is just the term we use for those 3 environments Tony was talking about. So RStudio, JupyterLab, SAS. But down here, there is a holder for custom interactive apps. So this is where you're going to start to see more and more shiny apps appear. Currently, they're all early access, but I'm mentioning them because they both have to do with GWAS. So there's one to access the locus zoom, and then there's also a Genesis null fitting a null model interactive shiny application. So if you want access to any of those, just email us, or you could simply email support via the question mark button on the bottom right. You can get early access to those. Okay, so if you want to use scripting, you go to Data Cruncher, and then you'll see a screen that looks like this. And the main thing is, if you're starting a new project, you won't have any environments that are running. But I've already started some because I wanted to demonstrate some things for you. And let me walk you through this. So I have 2 environments that are running. The first one is VCF visualization interactive analysis. So we're going to use that for quality control of our genotypes. And then there's the Genesis tutorial harmonization, RStudio notebook. And you can see in this table that they're different environments. So the VCF QC is JupyterLab, and it's running a specific set of programs, which I'll get into later. And then there's the RStudio environment, which is running an environment setup called Summer Institute in Statistical Genetics 21, because they used it last summer in their institute. So you can see that there's different environments, and they have different versions of R and different versions of Python. So this is important, and it'll come up later. So let me show you how to start one from scratch. So, but by the way, these 2 environments I cloned from the public project. So the concept is you have this environment, interactive analysis environment, that you can make copies of and move to different projects as you see fit. So if we want to make something from scratch, we hit create new analysis. And it brings up this create new analysis wizard. So I'll just call this JupyterLab. Log myself out of there. Back. Okay, so JupyterLab, let's call it Jupyter. And then you pick one of the 3 main environments. So JupyterLab, RStudio, or SAS. And under each category, you have an environment setup. And what's important about this is there's libraries that are pre-installed. A lot of bioconductor libraries, a lot of data science libraries are pre-installed, as well as if you're going to get into machine learning, there's the drivers that you need to do to run the libraries for TensorFlow, as well as the drivers to access the GPU. Okay, so we'll pick the default one, which is data science. And then you fill out your compute requirements. So here's when Tony mentioned, you can scale up. You can choose very large single node or a very small one, depending on what you want to do. It'll give you an estimate of how much per hour it will cost against your cloud credits. All right, and it sort of goes up from there.
Dave?
Yep.
I think you might need to reshare right now.
Okay. Yeah, thank you. Yeah, I got logged out. So what I'm going to do is reshare. Thanks, Tony. Log back in.
While we're waiting, does anybody have any questions at this point? Remember, you can put them in the chat or raise your hand, but this might be just a really good moment to just see if anybody has any immediate questions that we can cover.
There we go. That was good. Dave, now you're muted.
Okay, sorry about that, guys. I think we're good to go. Okay, so I was just saying that you can pick your instance type. It'll give you the amount per hour. You can get GPU instances. And notice I just know from experience that these are AWS instances because when I made this project, I selected the AWS platform. Likewise, I could have selected the Google Cloud platform if you're familiar with that or you're collaborating with other researchers that are using the Google Cloud platform. Of course, you can set a suspend time. So after your analysis is done, the system will go ahead and shut it down so you're not wasting any of your cloud credits. So I'm going to go ahead and start this up just to show you what it looks like. And it's going to initialize. So basically, you're getting your own private node. That's why it takes a minute or two. Along the top, you can go back to your project. You can stop it at any time, or you can access the help documentation or provide feedback. So you can ask, and that menu bar doesn't go away. So you can ask questions at any time simply by filling out this form. Our 24/7 support team will get back to whatever email address you have registered on the platform. So I'm going to go back. I'm just going to let that boot up, and we're going to use a different environment. So I just want to show you that you can actually have, multiple environments that have different purposes inside your project. And these are private only to the members of the project which you added. So let's start off with VCF. Let's start off with the Genesis tutorial. And as I mentioned, this was used in the Summer Institute that's put on by University of Washington. And if you want to learn about how to do a GWAS interactively, this is a really great tutorial. It's called the Genesis tutorial. It's an R Markdown notebook. But before we get there, if you remember, our first objective is to upload our own script. So I'm going to show you how to do that. In RStudio, there's a button. You just click upload. You can use this file picker to select any file locally. So I have a script called filter GDS, and I'm simply going to upload it. Text files are, scripts are just text files, so it's really small, and it uploads in a second. And this is a script that we have that I'm going to run line by line. And it's simply going to filter a GDS file. A GDS file is a file that's specific to R. It's a data object that includes your genotypes, and it's used in the Genesis workflow, so it's very important. But as I mentioned, the packages are already preloaded. So we have lots of bioconductor packages, such as seq array. And now I'm going to have the path be an object. So if you look here, there's really two locations for your files. There's the files that are hosted inside your project, also known as in your files tab. And then there's the files that are in this workspace. So in RStudio, you have the file workspace. And so the concept is you have this portable environment. So if I copied this and gave it to Tony to run in his environment, he would have all these workspace files, which are really the scripts, but he wouldn't have access necessarily to the files in my project. So that's why there is those two, and I'll mention this again later. But let me just run this interactively. So I'm going to grab the GDS file from the home directory in this analysis files folder. And I'm going to specify where I want to output my filter GDS. So in each, no matter if you're running JupyterLab SAS or this one, RStudio, you have a folder called output files. These are where you put your files that you want to put back in your project as soon as your session is over. Okay, so I'm going to open up the GDS file. And I'm going to filter for only the genotypes which are passing quality control and that are, and then the rest of the script that we're reducing the number of genotypes so to remove monomorphic variants. And you'll see things are getting done interactively. So I could change the frequency if I wanted to. I could do everything very interactive. And now I'm going to export, and I've exported this out. And before we move on, I want to mention, I'll probably mention this again in the JupyterLab, but there is a bash terminal that's accessible from RStudio, JupyterLab, and from SAS. So, and that's different than the console. The terminal is a bash terminal, which gives you access to install any type of software you want, but it also gives you access to this this folder structure, and everything under SB Genomics is important. So we have all of our project files. So if you have hosted TOPMed data, and some of it's open. So these are NHLBI, sorry, the TOPMed data is not open. You need to have the dbGaP permissions attached to your ERA Commons ID, but you can also access open data sets such as BioLink and 1000 genomes, which I want to show you here. We have, previously I added all those files under hosted data, and I copied over a bunch of these files. So you'll see chromosome 20, 21, and 22. I just want to demonstrate that there's that link that's taking place. So in the files, I have the same folder structure. So we have hosted data. So these are all the hosted. You can tell based on these icons that this is 1000 genome open data, and it's hosted in the NHLBI bucket. So we've made that link. So if I wanted to work on some of these files, I could as well. Okay, so we accomplished our first objective, which is uploading a script. Now, look into the future community hours. We'll describe how to take this simple script and turn it into a command line tool that can be made into a CWL app, let's say, that you can run and share with other people. Okay, so quickly, I'm going to show you some of this tutorial. So this goes through the whole process, but let's just take a look at phenotype harmonization. I'm going to clear my R environment out. And so basically phenotype harmonization can be done in RStudio using a package called dplyr, which is part of the tidyverse packages. And it's really great for data munging and comparing different sets of phenotypes. So let's imagine we have 3 studies that we want to combine together, study 1, 2, and 3. And the great thing about this tutorial, it has the test data already in the environment. So I have 3 data frames now, 1, 2, and 3. and they have the same number of observations, but as you'll see later, they're not compatible. And this really walks you through it. So imagine doing this with your own data. You could take notes and sort of do different types of visualizations. If you'll see, study 2 has capitals in the variable names, but some of the other studies do not. So if I showed you study 1 here, you'll see that it's lowercase. So if we were going to concatenate these together, we would have to rename study 2. And so that's the first problem you might have with phenotype harmonization. Another problem has to do with the data itself. So the summary R function will give you some of the 5 most important statistics when you're looking at data, which is the median, mean, and max. And what you can see here is that study 3 has a much lower mean and median than study 1 and 2. So why is that? You'd have to consider, and this walks you through what you might consider. You might consider that, this study is just containing all children. So that's why the height value, the mean height is much smaller. But we know through reading about our studies is that the study 3 was measuring using inches instead of centimeters. So right now we're going to convert study 3 to be compatible. And let's go ahead and run the summary again. So we'll see that the median and mean are much more in line with 1 and 2 now. This is a really great tutorial. I recommend everyone sort of as an action to go ahead and try to run through this. So when you concatenate, you may want to mutate and add a new value and determine that all the columns are the same. And then we'll use dplyr to bind rows. So we're not going to be joining, we're going to be concatenating. So we're going to have one table that is all the variables. And this is good enough for a simple GWAS. So this will be the phenotype information. We also need to have population structure as well as genotypes. So I'm going to go ahead and pivot to show you another environment to look at genotypes. So some people love RStudio and some people prefer Python notebook. So I want to make sure we cover all our bases. So let's go back to interactive analysis. And you'll see that actually we have 3 environments. You'll see the test environment Jupyter that I started is up and running. I'm going to go ahead and shut that down because we don't need it. But we're going to use JupyterLab environment. So I'll open this up. Oops. Oh. I think I had it open in another tab. I had multiple tabs open. I had it open in another tab and it was complaining about that. So remember, we cloned this from the public project. So each interactive analysis environment has 1 or more notebooks. In here, we have a notebook called VCF Visualization Interactive Analysis. Before we go any further, I want to mention that you can also access other types of notebooks and environments, including Julia. This is actually an older version of an older environment. That's why you'll see an older version of Julia and Python 2 still available. But you can access your Bash environment and you still have access to all your project files. So this is key. So if you're linking hosted data, you'll want to be able to first step is to link it to your project via the data browser, some other method, and then access the data that way. So remember, I put everything in the hosted data, and you'll see that we have the VCF, the 1000 genomes VCF here. The test environment comes with an hc.vcf, which is really just a small haplotype color output. So let's go ahead and make this a little bit wider. So this is a really great environment notebook that walks you through all the different types of visualizations that you might take a raw VCF and want to go through. So things like variant density, TS-TV ratio, the number of SNPs, the number of indels, variant quality, and then at the bottom, which we're not going to get to, is even variant annotation. But remember, most of the modules you'll need are already installed in these data science environments. But of course, if you have another package, you can do pip install or however you need to get it installed. So I'll go ahead and import the modules. And the main one that we're using is pandas and scikit allele. So this is a great way to take a VCF and make it manageable using pandas and other Python libraries. So, and this is probably the most important thing, is that if you're referencing a file outside of the environment, you should always look in this directory, SB Genomics, project files, and then the name of your file. So that's the path. And now we're going to take that input VCF and turn it into a data frame using the allele.vcf to data frame function. And we can do things like look at the first several rows. And so it makes it a much more tidy way to look at VCFs. So one of my colleagues created all these nice helper functions. So we have a way to determine what variant color it is, a way to calculate variant density, TS-TV ratio, base changes. And I just want to show you that near the bottom, basically it collects all these analyses, all these visualizations. And then let's see if I did it right. And then you can plot them here. So we have really a nice way to zoom in and out to look at these different features as well as variant quality. So the idea is, you would have your genotypes, 1,000 genomes or TOPMed data. You would look at the quality, determine if you need to filter any more, and then you would basically have everything you need to start to do your GWAS test. So you'll have the phenotypes, you have the genotypes, and then even if you need some, of course, you need the relatedness matrices. If you wanted the test data in the Genesis tutorial, there is some simulated phenotype information and simulated relationship data. So that's all I have, but we have plenty of time for questions. I will encourage you as a call to action to log on and to look at some of the public projects. As I mentioned earlier, there's a Data Cruncher interactive analysis public project. So even if you're not interested in GWAS, there are many notebooks which may interest you that are different types of omics, such as single cell RNA methylation, RNA microbiome.